The Africa Yam project has three main objectives, which we call outcomes. One is capacity building in the 4 countries that are participating in this project. The second objective is to use new breeding methodologies to capacitate the 4 participating countries to be able to use new breeding methodologies to fast-track their breeding program. The third objective involves the production of varieties and population of yam, new population of yam, to be used for the breeding purpose.
So this is Africa Yam 2017 crossing block here in Abuja. So we target 2 important species of yam, alata and rotundata, and our aim is to develop new varieties and populations for genetic study that fit to the outcome 3 of the Africa Yam project. And here we have the females, the rotundata on this side and alata on that side. So we have 24 rotundata, the white yam, and we have 12 female, the water yam, under crossing in this field. This is our male crossing block. We planted in isolation distance. So the main objective of our male crossing block is to collect these pollens from here to the female crossing block we planted in isolation distance. We use isolation distance to avoid any open pollination. So here we have both rotundata and alata, and we use isolation distance of about, this one is about 1 kilometer away from the female block. Okay, so welcome to our screen house here in Abuja. So this is under this screen house. When we get through seeds from the crossing block, we sow the seed here and generate from seed to seedling. So these are the seedlings coming from last year crossing block, and the main purpose of this seedling nursery is to generate from true seed or botanical seed to mini tubers that we can take to the field for further evaluation and selection to generate new varieties. Welcome to the 2017 Africa Yam Project Field Trial Site Abuja. So here we have different stages of trials. Once we get our seedling tubers from the screen house and we harvest tubers, and then we plant here as a tuber family evaluation trial. So this tuber family evaluation trial, the objective is to evaluate the families and also to select the best performing early generation clones. So we start single plant selection here. Once we plant single plant, and then we give identity for that particular clone, and then we move from single plant to row clone that we call the second clonal generation. So this side we evaluate these materials and so many traits, and then once we evaluate them, we again select down into a smaller number of clones, up to 40 and 50 clones, and then we put into a stand like that one where we have replicated a check that contains about 49 clones that include the standard checks and the local checks, and then we put into 1 or 2 locations. When we identify some good materials from this, then we move to multiplication trial. So here we have regional variety trial, in-country multiplication trial, both alata and rotundata, and we also set up separate seed plot where we put all our clones and evaluation in separate seed clones. Then we generate quality and a uniform seed for the next year planting. So this whole field is our selection and evaluation field, the 2017 near here in Abuja.